Okay, today's topic is the first one is PCR and the second one is hybridization technique. And I think we have stopped there in the identification of recombinants. Now let us take an example. Let's say a host cell which is having our plasmid which is containing our gene of interest. So we have actually identified a host that is containing a recombinant plasmid, but we are so far not sure that this particular gene insert is actually the one which has already been inserted by us or some other. It may be some other random pieces might have got inserted. So we use certain techniques like PCR, hybridization to confirm the presence of our gene of interest. They will tell us whether they will confirm the presence of gene of interest. Okay, the first technique is PCR, which is polymerase chain reaction. As the name says, it is done with an enzyme polymerase. Okay, now we can define PCR as it is the selective amplification of DNA. That means, suppose we have a gene of a DNA fragment, and say for example we need to make multiple copy of this particular region, which can be done with the help of PCR. This PCR technique is invented by Carey B. Mullis. This is in the year 1985, and this technique is one of the important technique in the molecular biology, and for this discovery Carey B. Mullis was awarded with Nobel Prize. Okay, now let us see how this is being done. See, actually the principle of this technique is complementary base pairing. You know, if we have one DNA fragment with us, with the help of an enzyme we can make the other particular complementary strand of it. Which is done by an enzyme known as DNA polymerase. Now this particular DNA polymerase mechanism is actually exploited for the technique PCR. There's 3 steps for PCR. The first one is denaturation, second is annealing, and third step is extension. Now we take the samples such as the DNA strand which need to be, or the DNA which need to be amplified. We will add DNTPs. DNTPs means all the required nucleotide phosphates, GATC. Okay, DNTPs. Then to this we add a special enzyme The name of enzyme is Taq polymerase. This Taq polymerase is isolated from Thermus aquaticus. This TAQ is actually it shows, or it is a reference of Thermus aquaticus. This bacteria is seen in hot springs, and these bacteria can withstand a temperature more than 100 degrees Celsius. So this enzyme can easily withstand that temperature. And now another important thing which is added in this is primer. As you all know, when a DNA is synthesized or polymerized, DNA polymerase cannot start from the space. They require a small fragment, and this is providing 3' OH end, to which they can add new nucleotides. So they require a primer, and this primer's 3' is actually extended by DNA polymerase. This we have already seen in DNA replication, class 11. So we give primer, and we have given DNA to be amplified, DNTPs, and all these will be done in a buffer. Obviously buffer will maintain the pH. And now what happens in denaturation, what happens in annealing, and what happens in extension? Let us discuss one by one. In the first step, that is denaturation, DNA which we supplied gets denatured, means the hydrogen bonding between A T pair and GC pair broken, and the DNA gets denatured. Actually this is done with the help of temperature above 80 degrees Celsius. Okay, and in the PCR machine actually we heat the DNA above 80 degrees Celsius so that they gets denatured. As you all know, DNA is having polarity 5', 3'. In the second step, the primer which we have already been given or added in this particular mixture gets annealed. Next step is actually annealing of primer. Annealing. This primer gets annealed at this end because DNA is synthesized always at 5' to 3'. So annealing, at the time of annealing, the temperature comes down to rapidly. We will have to reduce the temperature to 50 to 60 degrees Celsius. And during the process of third step, that is extension, temperature again we raise up to 70 degree or above 70 degrees Celsius, and then new DNTPs are added to this particular DNA strand, and again the PCR will start from first step. Now we got two DNA strands. They undergo first denaturation, then annealing, then extension. This continues, and very interesting thing is that after 30 cycles, more than 1 billion copies of our desired DNA can be made. This is the magic of PCR. Okay, PCR is used in a lot of applications. Why? Because this PCR amplifies the selective part. How it is being done? We prepare the primer which is based on this region. Then they will extend this particular region, and thus you can get many copy of that region. And PCR has done many wonders. You know, with this, because of this property, PCR is used in forensic science. In the crime scene, we'll get only little amount of DNA, and we can actually amplify it with the help of PCR. And now this is corona disease time. Now this corona is also COVID-19 virus is also actually identified with the help of PCR. Whether a particular region of DNA is present in that particular individual's secretions, nasal and throat swabs. If that particular organism is there, virus is there, that viral DNA part can be amplified, and we can see whether the individual is positive or negative. So disease identification, strain identification, then again fingerprinting may be PCR use karte. Okay, that's all regarding PCR. We'll move on to next technique, that is hybridization. So hybridization means hybridizing a probe to a DNA. So let us take this as DNA. A probe is a single strand labeled DNA. A single strand DNA, and they are labeled. Labeling can be done with the help of fluorescence or radioactivity or some color, so that if this go and bind to any part of our original DNA, we can dictate that region. Okay, this is in simple hybridization. So probe and primer. Probe is used in southern hybridization technique, and primer is used in PCR. Primer is also single stranded DNA, but they are not labeled, whereas probe is single stranded and they are labeled. Okay, let us see what is hybridization technique. Here we are studying southern hybridization. Southern is the name of scientist who discovered it. Okay, and southern hybridization is done for DNA. So you may ask, is there any northern, western hybridization, etc.? Yes, northern is done for RNA, western is done for protein, and there is no eastern hybridization. Only three types: southern, northern and western hybridization. In our topic or in our syllabus, we will be dealing with southern hybridization. So if you want to identify a particular sequence, you can also do by southern hybridization. First of all, we will have to take DNA, and this DNA are restricted by using restriction enzymes, and they will be separated on a gel by using agarose gel electrophoresis. Now this gel contain different bands which are actually having the DNA based on their size. This is positive end and this is negative end. So this DNA fragment will be of smaller size, and when you go up, the DNA size get actually increased. Okay, now this gel is a first part of hybridization. Then this gel is now kept on a nitrocellulose membrane paper. This is gel and the other one is nitrocellulose membrane paper. We make a copy of this gel on a nitrocellulose membrane paper, and this copy is done with the help of a technique known as blotting. Okay, so they can be done by various techniques like electro blotting by means of electricity, capillary force can be used, vacuum blotting is there. So by different techniques, what we do, we just transfer the DNA fragments from gel to a nitrocellulose membrane paper. Okay, with the help of electricity or with the help of capillary force, some DNA bands get actually transferred on nitrocellulose membrane. This is our gel and this is nitrocellulose membrane. Okay, now the DNA from this gets transferred to nitrocellulose membrane paper. Now the third important part is we are having now one is our gel and other is our nitrocellular membrane. Both will have same copy, same bands. Okay, now you can keep this gel in one side and we will start with nitrocellulose membrane paper. To this nitrocellulose membrane we will hybridize probes. Before hybridizing probes we will have an alkaline treatment. Why alkaline treatment is done? Just to make this DNA single stranded. Then only the probes can hybridize. And this particular gel is flooded with particular nitrocellular membrane is flooded with the probes. Later on this particular nitrocellulose membrane is dictated for the presence of probes. If probes are radio labeled, we can say by using autoradiography we can dictate the presence of probes on this particular paper. Suppose this particular part only got radioactivity, we can say that this second middle band is containing our gene of interest, because probes are made according to our necessity. Probes are having complementary sequence of our gene of interest. So among the three bands, middle band is containing our gene of interest. So these are the topics we discussed today. First one is PCR and the second one is hybridization. Both are used for confirmation of particular DNA sequences. Okay, if that DNA sequence is our gene of interest, then we can use PCR or hybridization for confirming the presence of those genes. Okay, thank you.